Hello all, welcome to Kathus Media. Today we are discussing about the chapter Sexual Reproduction in Flowering Plants. Parts of a flower. The parts of a flower are sepal, petal, then androecium and gynoecium. Androecium consists of anther and filament, together called as stamen, and gynoecium consists of stigma, style and ovary, also called as pistil or carpel. Pre-fertilization structure and events. Androecium and gynoecium. Androecium consists of stamens and represent the male sex organ, and gynoecium represents the female reproductive organ. Stamen. Stamen consists of two parts, long and slender stalk called filament and a terminal bilobed structure called anther. A typical angiosperm anther is bilobed and dithecous, and it contains 4 microsporangia. And the microsporangia develop and become pollen sacs, and inside the pollen sacs we can see pollen grains. Microsporangium or anther. Structure. A typical microsporangium is surrounded by 4 wall layers. They are epidermis, endothelium, middle layer and tapetum. The function of the first 3 layers is protection and helps in the dehiscence of anther to release the pollen grains. Tapetum nourishes the developing pollen grains, and it is multinucleated with dense cytoplasm. In young condition, the center of anther consists of cells called sporogenous tissue. Tears of young anther and enlarged view of one microsporangium with 4 wall layers, epidermis, endothelium, middle layer, tapetum and sporogenous tissue. Microsporogenesis. The process of formation of microspores from pollen mother cell through meiosis is called as microsporogenesis. Process of formation of microspores from pollen mother cells through meiosis. The sporogenous tissue of microsporangium differentiated into microspore mother cell or pollen mother cell. The microspore mother cell undergoes meiosis and gives rise to haploid microspore tetrad. On dehydration, the microspore tetrad dissociated to form 4 microspores. Each microspore develops into a pollen grain. Pollen mother cell is diploid in nature and it undergoes meiosis 1 to form 2 cells and later meiosis 2 to form 4 cells. A tetrad of 4 haploid pollen cells, and they secretes walls around them and they develop into separate pollen grains. Pollen grain. Pollen grain represents the male gamete of height. It is spherical and measuring about 25 to 50 micrometer in diameter. It is covered by 2 layers. The hard outer layer is called as exine and it is made up of sporopollenin, the most resistant organic material. Exine has prominent apertures called germ pore where sporopollenin is absent. Inner wall is called as intine. It is thin and continuous layer made up of cellulose and pectin. So outer layer is called as exine and inner layer is called as intine. Mature pollen grain has 2 cells, vegetative and generative cell. The vegetative cell is bigger and has large irregularly shaped nucleus. Generative cell is small and floats in the cytoplasm of vegetative cell. Economic importance of pollen grains. Pollen grains cause allergies and bronchial infections leading to respiratory disorders like asthma, bronchitis, etc. Example, Parthenium or carrot grass causes pollen allergy. Pollen grains are nutritious, hence used as pollen tablets as food supplements. Pollen consumption increase performance of athletes and race horses. Viability of pollen grain. Pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. The period for which pollen grains remain viable is highly variable and it depends on the prevailing temperature and humidity. In cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. In members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. Pollen grains can be preserved for years in liquid nitrogen at minus 196 degrees Celsius. Such stored pollen can be used as pollen banks similar to seed banks in crop breeding programs. Pistil or carpel. The gynoecium consists of a single pistil, that is monocarpellary, or may have more than one pistil, that is multicarpellary. When three or more than one, the pistils may be fused together, that is syncarpous, or may be free apocarpous. Each pistil has three parts: stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma, and the basal, bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity or locule. The placenta is located inside the ovarian cavity. Megasporangia arise from the placenta, commonly called ovules. The number of ovules in an ovary may be 1 in the case of wheat, paddy, mango, or to many in the case of papaya, watermelon, orchids, etc. Structure of megasporangium or ovule. Ovule is a small structure attached to the placenta, and the stalk of the ovule is called as funicle. The body of the ovule fuses with funicle in the region called as hilum. Hilum is the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus except at the tip, where a small opening called the micropyle is organized. Opposite the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials. The embryo sac of female gametophyte is located in the nucellus. An ovule generally has a single embryo sac formed from a megaspore. Structure of ovule or megasporangium. Hilum, funicle, micropyle, micropylar pole, outer inner integuments, nucellus, embryo sac, and chalazal pole is also seen in the structure. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called as megasporogenesis. One cell of the nucellus towards the micropylar end differentiated into the megaspore mother cell. It is a large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergo meiotic divisions, and the meiosis results in the production of 4 megaspores. Out of the 4 megaspores, only one become functional. Observe the diagram of megasporogenesis. Female gametophyte. In majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte or embryo sac. The method of embryo sac formation from a single megaspore is termed as monosporic development. Nucleus of the functional megaspore divides mitotically to form 2 nuclei, which move to the opposite poles, forming the 2-nucleate embryo sac. 2 more sequential mitotic nuclear divisions results in the formation of 4 nucleate and later 8 nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed by cell wall formation. Monosporic embryo sac development, leading to the formation of 8 nucleate embryo sac. 8 nucleate, 7-celled embryo sac. After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below this egg apparatus in the large central cell. Characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells at the chalazal end are called as antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity has 8 nucleate, 7-celled condition.